Hello everyone. Is my screen visible to you all?
Yes, ma'am.
Okay. So let's begin. All right. So before I begin, I want to ask, have you guys seen week 4 lectures? Why I'm asking is, if not an issue. Like I'll go in more detail about the questions. If yes, maybe simple points I can skip. Right. So that's why I'm asking. It's fine if you haven't seen. Just let me know if you have seen or not.
No, ma'am, I have not yet seen.
Okay. Alright. So what about Varsha, Sandhya? Have you guys also not seen? It's fine. It's totally
Yes,
fine.
ma'am.
You have seen?
No, ma'am.
No. Okay. Not an issue. All right. So um, just quickly, I'll introduce myself. I'm Ochandra and I'm a PhD scholar at IIT Kanpur, and I'm working in this area of tissue engineering. And every Saturday we'll meet at 4 p.m. All right. All the session is recorded, and you guys can access the video recordings in my YouTube channel. So have you seen that I have started uploading? All right, in my YouTube channel. The link is also given to you guys. If you have not seen yet, I request you to go to your portal, NPTEL portal. There you will see the problem solving session tab on your left hand side. There you will find the links of the YouTube videos. Also, I've uploaded the lecture notes of the past 3 weeks as well. Okay, so if you have any queries, you have to unmute yourself and ask. The chat option is not available. So quickly, we will recap of what is being discussed in week 4, or briefly, we will understand what is being discussed in week 4. In week 4, we are looking into what are the characterization techniques that are used to understand the properties of biomaterials. Okay, so you know by now that biomaterials here are we are using these biomaterials. Why? Because we want to make a scaffold or artificial ECM onto which the cells can be cultured. Okay, so that together the cells and the scaffold mimics your native tissue. Okay, that's why biomaterials is being used. Now, why it is important to understand the properties of the biomaterial? Because you will be able to understand then why the cell is behaving like that. Okay. Um, what didn't work? Where did you fail to understand the phenomena? So it gives a much more clear understanding of how the tissue is going to function in vitro outside your body, and how well you are able to mimic the properties of the tissue in lab with respect to your tissue that is found in your body. That is why characterization or knowing the properties of the biomaterial is important for us. Okay. So overall, we can uh, classify the properties of biomaterials into 4 types. So we're interested to know about 4 types of biomaterial properties. First is surface properties, mechanical properties, physicochemical properties, and biological properties. Surface properties talks about what is the nature of the surface. Why surface? Because it is the surface what that is seen by the cells. Cells cannot comprehend the overall bulk nature of the material, but it can understand the surface. That is why knowing about your surface properties is important for us. And as you can guess, the scaffold size is always bigger than the size of one cell. So cells will be able to feel through its receptors the nature of the surface, how hydrophilic it is, hydrophobic it is, what type of groups are there, the functional groups, yes, chemical functional groups. So those properties will affect how well your cell is going to behave. So predominantly it is the surface properties that directly influence the properties or the behavior of the cell. Okay. Then comes mechanical properties. So mechanical properties talks about bulk. Okay. So in order to clarify what is surface and bulk, let me draw a cube. Fine. Let's say your scaffold is of a cube shaped structure, a cuboid shaped structure. So here, surface is this. The red shaded portion is your surface, or this is your surface, or this is your surface onto which cells can adhere. Whereas what is bulk? Bulk is inside. Right? If we do a cross section like this, then a surface gets created first. This is your surface. And rest of the portion that is inside is your bulk. All right. The mechanical properties come from the bulk of the scaffold or material. All right. So we test how mechanically rigid it is. Then we also test if it is viscoelastic. If the material has viscoelasticity, then we also test the creep behavior, stress relaxation behavior. Okay. So depending on what the material is, like whether it is solid, liquid, viscoelastic, accordingly we have some characterization techniques that quantifies maybe the strength, fatigue, creeps, then stress relaxation. These are the way of quantifying or understanding the bulk property of a solid or viscoelastic material. Okay. Usually scaffold is not liquid. Then apart from bulk properties, we are also interested in knowing the physico-chemical properties. So physico-chemical properties include how, if it is a hydrogel, then we would like to understand how much it's going to swell or how much it is going to shrink. Then degradation, rate of degradation, how fast it is degrading. Then how much is the, if it is hydrogel again, how much is the gel fraction and how much is the water fraction or solution fraction. Then what is the porosity within the material. So these all comes in under physico-chemical properties. Then lastly, since this material is going to be interacted or going to be in touch with cells, right? So we are also interested how the cells are going to behave when in contact with this material. So those properties comes under biological properties. So we want to make sure that the material does not promote bacterial film formation, biofilm formation, means it should have antibacterial properties. Then whatever cells that you are going to add or culture on this material, it should stay alive. All right. And you want to quantify what is the percentage of the cells are alive over a period of time. So we do this using a class of tests that is called cell viability test. So these are some uh, characteristics, the classes of characteristics that we're interested to know about a biomaterial before we actually use it or actually show its usage for making an artificial tissue. Okay, we'll go over the questions one by one. And as we are going over the questions, I'll also try to uh, discuss about some theory behind this question. If you have any queries while understanding, let me know. Like in between, maybe I'll switch over a web browser where I can show you some images and all. Okay, because I've only added questions in this PPT because I had less time to prepare today. But like in between, I'll switch over to web browser and show you some images. Images will help you understand better about anything. All right. So this is the first question. The surface charge of a biomaterial plays an important role in cell adhesion and proliferation. Which of the following techniques can be used to characterize the surface characteristics of a biomaterial? One first is secondary ion mass spectroscopy. Option B is Fourier transform infrared spectroscopy. C is blood compatibility studies. D is Almar Boost assay. So these are the names of characterization techniques. Okay. So option A, option A, what is secondary ion mass spectroscopy? It basically talks about what kind of atoms are there on surface. Okay. So like I'm not very much familiar with the mechanism of the instrument that is used for this technique, but the why we use this technique is to understand what type of ions, atoms are present on a biomaterial surface. All right. So that helps us to understand what kind of charge will be there, if it is in charge form, then also what kind of atoms are there. So some atoms, it's like carbon based biomaterials, are generally found in our body, and as well as um, cells grow better. All right. But if your material has, say, some fluorine, all right, or like mercury, so these kind of atoms will be very toxic in nature. So in order to distinguish what kind of atoms are there, um, we can use this technique to do that. Then Fourier transform infrared spectroscopy, in short it is called FTIR. So in this technique, what we do, we try to uh, we first apply infrared light or infrared radiation on a molecule. So what this infrared radiation does, it causes vibration in bonds. So in that does what? It causes bonds to bend, to oscillate in symmetric or asymmetric manner. All right. So it causes some vibrational motion in the bonds, like say carbon-carbon bond, carbon-fluorine bond, carbon-hydrogen bond. So those in those bonds, some vibration takes place. So that is recorded. Okay, that is recorded in this technique, and we get something called FTIR spectrum. Okay, so I'll show you in a minute the FTIR spectrum, how it looks like. And what the FTIR spectrum tells us, it tells us about what kind of chemical groups are there in your compound, whether it is, and the characterization technique is also powerful, uh, meaning it can be used with either solid material, liquid material, all right, polymer film. So any form of compound or material can be used in this particular technique, and in that material we can come to know what kind of functional groups are there. So how FTIR spectrum looks like? So it looks something like this. All right. Here as I said, we irradiate the sample with infrared radiation, and some of the radiation is absorbed in form of vibration in within the bonds, all right? And whatever is transmitted out, that is captured. Whatever radiation is emitted out, that is called transmittance, that is captured by detector. And that detector basically record the transmitted light in form of this. It gives a output like this. Okay, this is called FTIR spectrum. So from the peaks, we can understand which peak correspond to which bonding. So here around 3500 wavelength, here we say that it is OH, right? Then here like around 1500, it corresponds to NH bond. So by stitching together, um, we can actually understand the chemical structure of our molecule. But please note, FTIR itself is not a sufficient characterization technique. It can help you validate, but it cannot help you prove. Okay, so let me repeat. uh, this technique is powerful when you know your original molecule, you have done some reaction and added some small functional group. You already know the structures of the functional group and your original molecule. So you want to just validate whether that functional group has successfully bonded with your molecule. For that application, FTIR is good. But if the molecule is unknown, means you do not know the structure of the molecule, then it's very difficult to deduce the structure of the molecule through FTIR. Right? So FTIR in that case is not sufficient. You need few more technique like NMR, okay, nuclear magnetic resonance spectroscopy. So to understand the entire structure of an unknown molecule. Okay, NMR is also widely used, by the way, for understanding the structure of biomaterials. Right. Then there is blood compatibility studies. So what are these studies? These studies comes under biological properties. The previous slide I showed. So they help you to understand whether your material can allow the blood cells when the material is in contact with blood. So your material should not be toxic to blood cells. The blood cells should remain unharmed in presence of your material, right? Otherwise, it's useless. Your body, everywhere there is blood circulation, and in wherever organ you are going to transplant, blood is the carrier of nutrients, oxygen. So the blood will get in contact with your material. So your material cannot be toxic to blood cells. Blood cells should be happy around your material. So in order to test that, we do compatibility study or blood compatibility study. This is Almar Blue assay is a type of again biological to test the biological properties. We do this assay. Here, what we do, we try to quantify the percentage of live cells. So we grow, we seed some number of cells, it grows, and we want to understand how many of the added cells are live or how many of those are dead. Okay, so greater the live cells, higher will be the viability percentage. So in order to find that, we use this assay. Now individually I have explained. I request to all to read this question again. Okay, what is written in 3 lines, and give me an answer to this question. The surface charge of a biomaterial plays an essential role in cell addition and proliferation. Which of the following techniques can be used to characterize the surface properties of a biomaterial? A, B, C, or D? Which option seems highly likely to be correct? Yes, Janani, are you able to hear me? Janani, can you answer? Vandana, what about you? See, if it were a physical class, it would have been difficult, right? To stay silent because I could see you. Now I cannot see you. Only I can read my screen and call out your names. But that doesn't mean you will take advantage of it. What seems correct? It's totally fine. If it's goes wrong, don't hesitate to answer. Okay, it will build your confidence.
Option A, ma'am.
Option A. Good. That is the correct answer. Okay. Then is the next question. Which of the following microscopy has a higher resolution? So here resolution, what do you guys understand about resolution? So we buy mobile phones, right? We try to get a very good screen in our mobile phones. People say should have very nice high number of pixels and very an AMOLED screen, OLED screen, this kind of screens we try to get. But why high number of pixels, like how it helps, why to spend so much of money in those kind of screens? Yeah, so let me ask this question in another way. So I'll quickly for a moment I'll stop sharing my screen. Okay, for a moment. Now can you guys see my screen? I've shown you two images here. Okay, this is first is image A, this is image B. Which among the two is better? Like which among the two is a better image? A or B?
Option B. B, ma'am.
B. Why?
That image was very clear, ma'am.
Yes, it is clear. Why? But why it is clear? What is the right term to use here? This image has a higher resolution. Okay. So if you take a cube of same size in this image, then what you'll see is, let me quickly go back to my PPT. So So if this is your square, this is your square you have taken from image image A. This is the square you have taken from image B. Here, the number of pixels that is there maybe 300 that can fit into this image B. But here, the pixels that can fit in image A, maybe it was 50. So when it was magnified, the image was less clear, it was blurred. Its resolution is not as good as the resolution of image B. Now, this is a very common problem when you capture an image using a poor camera. All right. Maybe in 2013-14, the mobile phones that used to come that time, those mobiles have a very poor that will like very give very poor resolution image in comparison to what we have today, right? Camera wise. So similarly, what is microscope? Microscope is nothing but we are trying to capture an image through microscope, but at a lens scale much smaller than you can see with your naked eye. If we were to if that object was observable through naked eye, you don't need microscope for it. But if it is very small in the micron scale, then you need a microscope. Okay, so these four techniques, sorry, these three are type of microscopic techniques where we try to capture image in of some good resolution. I'm not going to tell you which is having the high resolution for now. Only difference in these three type of microscopy is the source of light. You know, for capturing image, good lighting condition is required. Similarly, in microscope as well, when lighting, some source of lighting should be there to illuminate your sample, whichever sample you're trying to take an image of, or whichever object. So here in scanning electron microscope, we use an electron beam as the light source. The electron beam falls on your material, all right, and then we capture image. Okay, wherever it is falling, we can capture image at different magnification. We can go to a very low magnification to very high magnification. In fluorescence microscopy, what we do, we give a monochromatic light. Okay, what is monochromatic light? Light of a single wavelength. And that wavelength of light helps in uh, illuminating some fluorophore. Fluorophore or those compounds that can fluorescence, emit some fluorescent light. Those are called fluorophores. So in presence of a light of a particular wavelength, your material containing some fluorophore will get illuminated and you will get some colored, brightly colored images like blue, green, red, this kind of images you will get. Whereas in optical microscope, usually we use white light, simple white light, which is having all the wavelengths of visible light that we use. Okay. Now, only difference is that. Apart from that, there are magnifications possible. Okay. But the type of magnification depends on the lens you have. So there is 100x magnification possible that can zoom in even up to 1 nanometer. Also, there are 4x magnification possible that can only allow you to see up to 1 mm lens scale. So depending on what objective you use, you can zoom in and zoom out. Okay. So just a brief idea about what these microscopies are and what are the light source. Now, can you guess which of this microscope can give you better resolution? You understood what is resolution, right?
Yes,
What do you
ma'am.
understand? Yeah. No, just simple guess. Which microscopy can give you better resolution images?
Electron, ma'am.
Electron, right. So it was a good guess. It is electron microscopy. In fact, electron microscopy can go up to few Armstrong's order, means 10 raised to the power minus 8 Armstrong. Whereas fluorescence or optical microscopy, they can only go up to millimeter order, means 10 raised to power minus 3. So here, what this means, Armstrong, it means if you take same square, all right, there are more number of pixels. So each of these images are going to give you an image. So about that image I'm talking about. So the images that you will get from electron microscope, you will see more pixels. The image will be much more clearer. All right. Whereas for fluorescence microscopy or for optical microscopy, if you zoom in, you will see the image will start getting blur and blur. All right. So let me show you some examples of how the results look like in electron microscopy, in fluorescence microscopy, and in optical microscopy. So this is how some of the images looks like. You see how detailed the images are here. Even the spikes and the dots, very detailed in nature. This is how the SEM image or electron microscopy image looks like. Here you can only capture image of this surface. All right. So this is another image here, right here. Here's some cells or some maybe bacterial rods. Those are interlinked. So very clear, very fine images can be captured. For fluorescence microscopy, you see, I told you how colored images you can get. Here, fluorophores are used, all right? So they have captured the image of a cell, all right? The cell, within the cell, there is nucleus and there are some proteins like actin, vitamin. So these proteins and this nucleus, they are tagged with different fluorophores. So one fluorophore is giving a blue color. So that is how they're distinguishing the nucleus. Some fluorophore is giving red color. So it is getting distinguished using actin. And here you're eliminating your sample with one wavelength light or monochromatic light, which actually causes fluorescence to occur in your fluorophore. Okay. This is how a fluorescent image looks like. So if you, this is an image of how optical microscopy will give you. So they have captured one thing at different magnifications. It is not as beautiful as the fluorescence and the SEM images looks like, but this is how they look like in general. It can help you see the structure of cells. So let me show you. So this is uh, how the cells look like under an optical microscope, individual cells and as well as group of cells. So at different magnification, you can capture it. Okay. So these are some examples, and I hope you understand better about the microscopy technique that is mentioned here. Now, this is the next question. Which of the following techniques consist of a cantilever with a probe at its end that scans this surface or substrate roughness? Okay. So again, like briefly, I'll describe each of the techniques that is mentioned here. So do you guys understand what is a cantilever?
No, ma'am. What is cantilever, ma'am? I don't know.
Okay. I'll show you with an image. you see the weighing balances, right? जो like with a stand, ऐसे stand ऐसे बना रहता है weighing balance का. एक second. तो कैंटिलीवर में क्या होता है, one end of a plank suppose is fixed, okay, and you apply some load on the other end. So it can basically, when you apply some load, it can directly go back to its initial position. So this is a very primitive explanation of how a cantilever looks like. It is a very well explored concept in civil engineering, where they use this concept to make bridges, then in houses, right? They create some outward roofs, right? So they are trying to actually create a cantilever structure. Now, cantilever with the probe will be like this. So one end is fixed, as I told you. One end of the beam is fixed, and the other end, there is some fine tip, very fine. It can pierce you, something like that. That tip is there. So as the uh, beam or this cantilever it can move, right? Because one end is fixed only, other end is free. So it can move. This helps uh, probe and this cantilever together, it helps in moving. And there is a detector connected at the end of the cantilever that monitors the movement of the probe. That is, this is how a cantilever with the probe looks like. All right. So this probe can move over a sample. It will move as the sample surface is moving. The sample surface is rough, the probe will accordingly make its path. If the And it can also allow other measurements, like it can also help you understand the stiffness of the surface. So if the material is very stiff somewhere, the probe will not go deep into, right? If the surface somewhere is very soft, the probe will pierce, and the movement of the probe is held with this cantilever structure. And then end, there's a detector that captures the motion of the probe. All right. This is the probe, cantilever with the probe. This is how it looks like. Now, these are the techniques that are mentioned here in option A, B, and C, where in one of these techniques uses this cantilever with the probe to scan the surface roughness. One of these techniques uses that. First technique is called surface plasmon spectroscopy. B is Raman spectroscopy. C is atomic force microscopy. D is traction force microscopy. Okay. So if any one of you has seen the lecture, can you answer which of the techniques work with the cantilever and a probe? Okay. The first technique that was mentioned was surface plasmon resonance spectroscopy. So here what is happening? This is a biomaterial, all right? And on top of it, there are some like molecules present, some functional groups or something like that. Those are present. We emit some light source, and the transmitted from the surface, it is captured by detector. And what it helps in understanding? It helps in understanding how the material is like reflecting. So just give me 1 second. I can show you a better image maybe. So this spectroscopy, as it mentioned here, look here. It's a biosensor based method that observes the interaction of a protein in the mobile phase with specific site immobilized at the sensor surface. The detection principle is based on the changes of the optical properties of a surface layer with increasing mass of absorbed protein. So, yeah, this is your surface. And on the top of the surface, you have conjugated some antibodies, suppose. Okay. And these antibodies have covered some area of the surface. So it is reducing the transparency of your surface. As more and more antigens are present in the area, they will go and complementarily bind with the conjugated antibodies. Then what will happen? The again your reflectance means more less and less light will now reflect back. The surface has become much more denser. So the in this way, indirectly, by change in the optical properties of your surface, your conjugation between antibody and antigen is taking place. So this type of characterization of surface property is called surface plasmon resonance, that indirectly measures through change in optical property the protein adsorption on the surface. Okay. Then what is Raman spectroscopy? So Raman spectroscopy is actually a very complicated concept. It comes from a principle of Raman scattering. So there's a sample, you illuminate that sample with some laser, all right, and due to the power of that laser, the sample start scattering the light, and this scattering happens very randomly. That is called Raman scattering. So there's a detector that can actually capture the light that has scattered due to Raman scattering. And as a result, it can help you analyze what are the functional groups present in your compound. So it gives a spectrum like this, Raman spectrum it is called, similar to FTIR spectrum that I showed previously. So this is a Raman spectrum for paracetamol as well as aspirin. So depending on what kind of chemical bonds were there and how those bonds caused Raman scattering, there's a detector that captured the scattered light and gave you this kind of a spectrum. So this is again a signature spectrum of a particular molecule. Okay. Now, many information can be extracted from Raman spectroscopy. Different types of machines are also coming nowadays, but the primary objective of this technique is to find the functional groups. Now, next technique that was mentioned in that list is this AFM, that is called atomic force microscopy. So here, what is happening? Here is a fixed substrate like a blank or something like that, and on tip of it, on the end, one end of it, there is a tiny tip. Okay, and the other end is connected to a fixed surface. So one end is fixed, another end is free. So the free end can actually move over the surface, and as it is moving, the movement is getting captured in form of an image. That is called AFM. Okay. So some AFM images will look something like this. So this is your uh, plank. I'm not calling it with the original name right now because I want you guys to answer and later on, like understand and later on answer. And on tip of it, there is a tiny needle kind of a thing. It will move, all right, over the surface. And these are the detectors that can actually monitor the movement of the tip over the surface. And as it does, it captures and images, beautiful images of the surface. Here, wherever the surface is bit bulgy, that image was captured. Wherever there was a depression, that image was also captured. So this is how AFM works. Now, please note here some piezoelectric material is used. Piezoelectricity means the material that can change any mechanical force into electrical force. Okay, or electrical signal here. Okay, mechanically the tip is moving over the surface, that is converted to electrical signals, and that is being read by the detector. All right. And there is one more. I think by now you know what the answer of that question was. Question is, there's one more microscopy, traction force microscopy. So this particular microscopy is very useful when you want to understand how the cell has actually spread over a surface. Okay. So there are, so when the cell moves on the surface, the cell generates what is called traction forces. And this microscopic technique can actually quantify the traction forces and thereby can help you understand the morphology of the cell on a surface. So, yes. So this is the cell, right? And here are some traction forces generated. So the red regions in this image are showing where the traction force is the highest. Means at the pseudopodia region, the traction forces are the highest, whereas in the center, the traction force is the minimum. So in this way, we can capture the morphology of the cell here, right? And how it is generating traction force in order to move forward. It is a very important force that cell generates for migration. Okay. Now going back to the question that we originally had. This was the original question. Which of the following techniques consist of a cantilever with a probe at its end and scans the surface roughness? A, B, C, or D? I've shown you images, right, of each of these microscopic techniques. And did you see anything in those images that look like the cantilever with the probe? Anyone? Divya, what do you think is the correct answer here? Vandana, Okay. Anyone? Am I audible?
I think it's option C.
Option C. Yes, you're correct. It is atomic force microscopy where we did see in the images where they were using cantilever with a probe, and the images that it gave beautifully captured the surface morphology where it was depth, where there was some trough. That means it beautifully captured the surface roughness. Okay. Now, according to Hooke's law, what is correct here? Anyone? What is Hooke's law?
Option B.
Option B. Yes. But what about others? Familiar with Hooke's law? Hooke's law basically says that stress is directly proportional to strain. Right? So if we draw a curve, stress versus strain, the Hooke's law says that there will be a straight line around 45-degree angle that shows the stress is directly proportional to strain. So this is also called elastic region or Hookean region. Okay. So Vandana is correct. Option B is correct here. Stress is directly proportional to strain. That is Hooke's law. Now, the ultimate strength of a material is 50 kilopascal. What is the maximum load that the material can withstand? Do you guys understand what is ultimate strength? Anyone? What is ultimate strength? So I'll try to draw a diagram. Now in that diagram, tell me, okay, where which point in that particular curve will you say that it is ultimate strength? So the curve will look something like this. This is your stress. This is your strain. This is your typical stress strain curve of a material, of a solid material. So I'll give some points somewhere. Okay. So these are 3 points. 1, 2, 3. Which of these points in this curve correspond to the ultimate tensile strength or ultimate strength?
Point number 1, because after that it is the plastic region where material undergoes deformation.
So um, I like your confidence, by the way. But point one is actually the point where the material starts yielding. Means it is the point where the elastic behavior ends. Okay, so this is also called yield stress or proportionality limit. Means this is where that stress versus strain behavior stops. The entire this region is now the plastic region. Okay, so point number one is not uh, said as ultimate tensile strength. So this is a curve of tension. We are taking a material like this. We are providing tension. Okay, that is why it is this is a curve for tension. For compression, the curve will look slightly different. Now, now what do you think? Which is the point for ultimate tensile strength? 2 or 3? Ultimate tensile strength. Even okay. So I'll answer for you. This point 3 represents the point in the experiment when the material underwent fracture. Means the material broke in two pieces. That is why stress and strain values cannot be recorded anymore. Whereas point 2 represents the highest point in the curve, or it also represents the highest value of stress that the material could sustain or it could resist before it underwent fracture. So this point 2 is actually called the ultimate tensile strength. So if we were to define what is ultimate tensile strength, it is the highest or the maximum stress the material can endure before it fractures. That is ultimate tensile strength or ultimate strength. Okay. So in this question, what is mentioned here, that a material's ultimate strength is 50 kilopascal. Remember, it is the highest strength. So what is the maximum load then that the material can withstand? A, B, C, D. That kilopascal means 10 to the power 3. Right. Megapascal means 10 to the power 6. Anyone? So if this is the highest stress that the material can endure, then we cannot exceed it, right? We cannot exceed this value. If we go above this value, it will immediately break. It will break much earlier, means it will fracture much earlier. So if we are going to use this material in a load bearing application, we have to ensure that the load that is applied, it should be less than this value, so that that material functions for a very longer period of time. So which of these values in A, B, and C is lesser than this? Let me know where which part you are not understanding. Maybe I can try to reframe my explanation in better way. But I have to know where the gap is there. You are the one who can tell me. So which of these options A, B, or C, D is A, B, C, D has a value less than 50 kilopascal? This is 10,000 Pascal, meaning 10 kilopascal. All right. This is 5 lakh pascal. So if we were to convert it into kilopascal, it will correspond to 500 kilopascal. This is in megapascals. So if we were to convert to kilopascal, then it will be 650 kilopascal. This is already in pascals. So what is the maximum load then the material can withstand? Can the material withstand 600 kilopascal? Yes or no? Are you guys there? I totally understand that if you're feeling hesitant, please try. I request you to try, to think over the question and try to answer. The correct option is D. Okay. The maximum of this values that the material can endure is 40 kilopascal. 10 kilopascal it can easily endure, all right, but it cannot endure 500 kilopascal or 650 kilopascal of load because it will fracture immediately. So among this 40 kilopascal, it can endure the maximum value. It is slightly less than 50 kilopascal. Okay, is it clear? Yes or no? Is it clear? Is this question clear? So, Sugantha, is it clear? Vandana?
Yes, ma'am.
Good. Here is another question. Determine the degree of swelling for the hydrogel sample. So we have to calculate the degree of swelling. So how do you calculate degree of swelling? We call, I guess week 1 it was discussed. Degree of swelling is given by wet weight minus dry weight by dry weight into 100. Okay, so this is how you will calculate the degree of swelling. So in the question, we are given the weight of dry hydrogel, it is 150 mg. So wet weight, we don't know, but dry weight we know. It is 150 mg. And the weight of swollen hydrogen after 48 hours is 2000 mg. So this is the value of wet weight. Now we have to just fit these values in this equation and we'll get the degree of swelling. Okay. So how will we do that? If you're doing in your rough notebook, let me know. So what's the value you're getting? What's the degree of swelling you are getting? Take a rough notebook, a rough page, or maybe a mobile phone and fit this formula. What's the degree of swelling you are getting? Yes. What's the value you're getting? Are you guys doing it? Okay. So the value that I got is around 12,000, 1,233. Okay, when I fitted this equation in my formula, this is the value I got. I hope you also got the same value. Now moving on, the porosity for the scaffold is vital as it helps in cell attachment and nutrient transfer. So which of the following techniques cannot be used to measure the porosity of the scaffold? So porosity is a property that comes under physico chemical property of a scaffold. Porosity, the degree of porosity also affects the swelling degree, okay, in case of hydrogens. So these are uh, some techniques, characterization techniques. Only one of this cannot be used to measure the porosity of the scaffold. Briefly, I'll go over three images. What are these techniques? So first is gas pycnometer. So you guys understand what is porosity? These are nothing but some void spaces. All right. This is how the instrument looks like. Here, what we are doing inside this instrument, we keep our sample inside the instrument and we flow some inert gas through the sample. So what happens that if the material is porous, within the pores, the inert gas can enter. Then we allow the process to happen for some time until it reaches equilibrium. And then another gas is flowed again through the sample to remove the whatever air that was trapped inside the pores, and then it is captured through a detector. So the difference, the initial sample with air and sample without air, inert gas, as I said, inert gas is used to fill the pores. That helps in determining the void space inside the volume of void space, total volume of the void space inside the sample. Here the gases that is used to fill those void spaces, the volume of those gas, it helps in determining the volume of the void space inside the sample. Okay, this is this technique is called gas pycnometry. We are using a gas to fill up the void space and then quantifying how much volume of the gas was initially used to fill up those space. Okay, that basically is equal to the volume of the void space, a volume of porosity. Then another technique that was mentioned was scanning electron microscope. So you already know what is scanning electron microscope. Here we are just scanning the surface of the scaffold and trying to capture images, okay, of the scaffold. While doing so, we can also see some locations where which are very dark, which are, and there are also some subsurfaces which are brightly illuminated. Usually, the dark surfaces considered to be troughs or void spaces because light cannot pass through that depth. So that is why that void spaces comes up as a bright, dark region, darkened region. So SEM can also help in measuring the porosity. Then this technique that is called liquid displacement method. Here we actually use liquid mercury. So, so we dip our sample into a liquid solution. So mercury has been widely used for this method. The liquid can once immersed, the liquid can go and uh, fill up the void spaces. And then we compare the weight of the sample with liquid and without liquid. So if the liquid has entered, it will definitely means that it has gone into the white space because there is no other space for the liquid to stay. And the volume or the weight of the if you subtract the sample without if we subtract the weight of the sample with mercury with the weight without mercury, we'll get a difference of the amount of liquid that has entered. So that can actually help you understand. So this much weight or this much, if you divide it by density of, if we know the density of mercury, then we'll also know the volume of that void space. So this is another method generally used to understand the total volume of void spaces or porosity. Okay. Then the last method that is called captive bubble method. So again with an image. Image actually helps in this in these kind of characterization techniques. Image are widely helpful to understand the concept. So here, what we are doing, we have a surface, all right, and onto that surface here they have put some oil, and the contact angle that is being formed between the oil and the surface is measured. So this is a method actually to measure the contact angle or to determine the wettability in this particular uh, what is setup. Okay, this is a type of a setup to measure the contact angle. So among these four options, only option D is correct. Why? Because this is a type of a method to measure contact angle. All three options A, B, and C can be used to determine the porosity of the scaffold. Okay. Next. Next problem will go. If any queries, let me know. All right. What are the problems caused by bacterial biofilm that results in implant failure? So bacterial biofilm, when the biofilm is formed, what happens? It is very difficult to now target these bacterial cells with antibiotics. It is like it forms a tightly, the cells are tightly adhered and the cells are protected by a gelatinous film. Okay, so overall the structure is called a biofilm. And if any antibiotic, if you deliver to enter the cell, so there is a barrier of gelatinous film that stops the or reduces the effectiveness of the bacteria. So in the when the bacteria has formed biofilm, it is very difficult to treat it, or any bacterial associated infection. It is very difficult to do that. So once the biofilm is formed on your implant, what problems can it cause that will ultimately result in malfunctioning of the implant or ultimate failure of your implant? So first option is infection. It can cause infection? Yes, it can. Inflammation due to endotoxins caused by the produced by the bacteria, definitely. So when the bacterial cells are there, so bacteria are very well known to produce some toxic molecules that is called endotoxin. So these endotoxins are seen as foreign agents. So whatever immune cells like macrophages, the monocytes, etc., they will cause a inflammatory response at the site of biofilm formation, because of which that site becomes vulnerable, and ultimately it will not allow the implant to function well. It will also result in discomfort to the host. So that is definitely the problem that can be associated to bacterial biofilm formation. C, uh, can it cause donor side morbidity? What is donor side morbidity? It means the donor side is affected. So when we are doing, say, skin transplantation, people usually take it from the person itself onto which the skin is supposed to be transplanted. Suppose in case of a burn victim, people or the surgeons take a small section of skin from his or her thigh, and then transplant it on the burned site. That is called skin transplantation or autologous skin transplantation. So in those cases, what we are doing, we are actually causing injury on the donor's body. Means we are creating a new injury while doing so. So now in this case, when the bacterial film has formed, is it causing donor side morbidity? No, right? Like, yes, the site of the implant is getting affected because of inflammation and infection, but additional site in the body, they are not getting infected. Rest of the parts of the body, they are healthy. So that is why the correct answer here is both A and B. A and B are correct here. Okay, is it clear? I'll Since you guys are not responding, I'll assume that it is clear. If you have any doubts, I request you to please unmute yourself and ask. Then this is the next question. Cell migration and proliferation, they're necessary to accelerate the wound healing process. All right. So wound healing is a process that undergoes in our body only at the instant when your body is injured. Suppose you get a cut somewhere in your skin. So in order, since the barrier, skin is a barrier organ, right? It has gotten compromised. It has like opened up. So in order to close it, close the barrier, a a very detailed and elaborate cascade of reaction where multiple cells are involved in a phase-wise manner starts. That is called wound healing process. So the ultimate objective of this process is to close that cut as quickly as possible. So you must have seen some clot starts forming, and over a period of time, when you get the ick to scratch that clot, you will see new, very pinkish flesh of skin has started forming. And after a few days, that cut is completely healed. And what you see as a result is a scar. That is the result of the wound healing process that has occurred uh, at this site, which you don't know, but the cells are doing it, okay, in order to protect you. So for the wound healing process to occur, though two important, very important cellular fate processes that must occur is cell migration and proliferation. Okay, more and more cells should come at the site, whether it is immune cells or in skin. In case of skin, we're talking about keratinocytes, more and more keratinocytes, then blood cells. So all those cells should migrate at the site and they should increase in number in order to heal the wound. Now, which of the following methods can be used to study if the fabricated biomaterial promotes cell migration? Okay. So these are the some techniques used to characterize the biological property of a biomaterial, which I have mentioned here. One of these technique is very um, crucial to understand whether your biomaterial can allow cell migration on its surface. One of these technique helps you do that. So I'll go over one by one and then I'll tell the answer. The first is the MTT assay. So MTT assay is a assay that helps us to quantify cell metabolism, and indirectly you can also get to know cell viability indirectly, okay, not directly. Why I'm saying indirectly? It is coming from the logical fact that if the cell is able to metabolize, okay, if the powerhouse of the cell, the mitochondria, is functioning, it is an indication that the cell is alive. Okay, that is why I'm saying. So there is a chemical called MTT. The short form is MTT. If you search in Google, it's a very long name. So it's a type of a salt, and that salt can be reduced to form a purple colored compound in presence of mitochondrial enzymes. So mitochondrial enzymes will be only secreted in live cells. If the cell is already dead, the metabolism has stopped. So in those cells, these enzymes will not be present. So in presence of those mitochondrial enzyme, this compound is reduced to a purple colored compound. And depending on the intensity of the purple color compound, we can quantify how much cell viability is there. What is the percentage of cell viability? Okay. Then this is called, next assay is called Almar Blue assay. It is very, very similar to MTT assay. Only difference is the type of compound that is used, but it works, or the result that it will give is the same. It will also help you to understand the cell viability percentage. Okay. This is called another assay that is called scratch wound assay. So I'll show you an image related to this. So here what you see is a monolayer of cell in A. Okay. You use a very fine tip needle or a plastic, something very fine tip, and you create a line scratch onto the substrate. And what you do, you then leave it as it is. You observe how the cells on both the edges, so this on both the edges of the scratch, how the cells are going to now react. So what happens if your biomaterial supports cell migration smoothly? In panel E, you see the last row. You'll observe the last row. Suppose this is your biomaterial in the last row, but the material supports cell migration. Then eventually, over some period of time, you will see that the scratch that you have created, it will start disappearing, because the cells at both the ends of the scratch, they will proliferate and migrate. Okay. And slowly it will completely like make the scratch disappear, and you will see again a monolayer of cell. So this is called scratch wound assay that we do in vitro after growing some number of cells on a smooth surface like a petri dish, a glass slide, or something like that. Okay. And then we do this assay. So this is a very nice image. They are trying to say that over a period of time, the what do you say? The length of the scratch is decreasing. Indication that your material is promoting cell migration. Okay, onto the surface. Then another study often used is called co-localization study. Here they try to see whether the uptake. So uptake means you introduce some foreign particles inside the cell. That is called uptake. Maybe it may be a drug particle or nanoparticle. So we try to see if more and one or more such particles are present inside the cell, and then we quantify it. So that is called co-localization, and this technique is not usually used for cell viability or something like that, but to quantify some uptake studies. Uh, so yeah. So now, if you want to try, I'll give you a few seconds. Can you now answer which of these option is correct here? I've, I've straightforward given you the answer, but I still want to see if you have listened to my explanation carefully. If yes, it's a piece of cake for you. Okay, no response is fine. The correct answer here is option C. The scratch wound assay actually helps us to understand whether or not cells can migrate onto a biomaterial. Because as I showed you, over a period of time, that scratch disappears. So how it is disappearing? It is disappearing only if the cell will start migrating, right, towards like from both ends and start proliferating to uh, make up for the lost cells. So when that happens, then only the scratch will disappear. So in this way, you can quantify it or not. Now, this is a graph shown in your right hand side. In the graph, you have to tell what are point A and B called. This is a graph. This is a typical graph that you generally obtain from thermal property characterization techniques like DSC, differential calorimetry, or TGA, thermogravimetric analysis. So these are some techniques that helps us to understand what's the melting point, what's the glass transition temperature of your material. So you can understand then in presence of physiological temperature, whether your material can undergo physical changes, like can it um, like lose its structure or starts degrading because of heating, thermal degradation, can it happen? So these kind of questions can be answered when we do this kind of thermal property characterization techniques. So this is the typical graph that you obtain. So point A here and point B, you have to answer what is it. Notice point A, above point A, the material is in a glassy state. What is glassy state? It means it is in crystalline state. If it is a polymer, the polymer chains are very well arranged in a ordered manner. But as we're increasing the temperature, notice the x-axis, the state of the material is changing to a rubbery state. Okay. And as we are increasing the temperature, the polymer is have become a solution. Okay, so liquid flow or liquidity has come. It's like a it's like water, very viscous solution. Okay, it is no longer a solid anymore. And in between the glassy state and the rubbery state, some plateau all often seen. Temperature remains constant and then it starts like falling. So now for this dotted line, here also similarly, this first plateau at a lower temperature represents to a glassy state. Then as we increase the temperature, the material changes here to a rubbery state. But here note the plateau is missing. The plateau is not seen for a large temperature range. And here also liquid flow after at a very high temperature, it will transition to a liquid. Okay. Now, what this temperature is called? So A correspond to some temperature, B correspond to some temperature. So A and B marks those temperature where the material is changing its phase. If you see carefully, at A point, at temperature that is lower than A point, the material is in glassy state. And the temperature, if it is greater than A, the material is changing into a rubbery state. So that particular temperature is called something. Okay, I'll not tell you now. But here in B, point B, this is your glassy state. And if you see, the rubbery plateau is not that much. Okay, rather the material is transitioning, shows a transition to directly towards a liquid state. So from the glassy state to liquid state is happening in this particular curve. So what this temperature will be called then? And also note B is greater than A, right? Now these are the options that are given to you, A, B, and C. So clearly C and D is not correct, right? Here we are not talking about yield stress or elastic modulus or fracture toughness. C and D are not correct. It is either A or B. Anyone would like to try? So the correct option here is actually option A. Okay. As I told you, A here below A or below glass transition temperature, the material is in glassy state. And as we increase the temperature above glass transition, it reaches to a plateau region or a rubbery state. Whereas B corresponds to melting point or melting temperature. So as the term suggests, melting means the material is going to melt from a solid state to a liquid state. And B or the melting point is always greater than glass transition temperature. Okay, is it clear? Okay, I'll assume that it's clear. This is the next question. The surface free energy of a material can be detected using atomic force microscopy, scanning electron microscopy, transmission electron microscopy, and goniometer. So you by now you know what is AFM or atomic force microscopy. You also know what is SEM. Okay. The other two techniques, that is transmission electron microscopy. Here in C, what we do is we basically try to see through the sample. Okay. In transmission electron microscopy, we see through the sample. So this is the sample and the electron beam is passed through the sample. This is a very powerful technique. At even higher resolution, if we go using a very powerful or high voltage beam, we can see the atoms as well in transmission electron microscopy. And also we can see the mitochondria of a cell. This is a very, very powerful technique that can tell you about the structure which is inside the bulk as the material, as the beam is passing through. Goniometer is basically the instrument used for measuring contact angle or quantify the wettability. Okay. So now here the question is, we have to tell which of this technique can be used to measure surface free energy. So for surface free energy, if you take some time later on today or maybe anytime, try to look up what is surface free energy. Surface free energy tells you about, so there are some atoms, okay, on the surface. Okay. Now this is another layer of atom. If you see the atoms, if you see this interface, only one only one side of the atom is directly bonded to the atoms which are below it. Whereas whatever free electrons that are there on this side, upper side, they are free in nature. So because of this, unstable energy starts building up on the surface. And this is a unique property only of a surface. It cannot happen, or you cannot observe this property inside the bulk. All right. So surface free energy also give rise to another quantity that is called interfacial tension, a surface tension. If the substrate has a higher surface tension, then it will like affect the morphology of the droplet, or it will affect the contact angle as well. Okay. So now tell me which of these techniques can be used to measure or quantify the surface free energy or indirectly the surface tension. Surface tension is related to contact angle. This equation given, that is called Young's equation. So there we quantify the uh, interfacial surface tension or surface tension at different phases, like liquid solid interface, solid liquid interface, solid vapor interface. Okay, using the help of the contact angle between the water drop and your surface. So the correct answer here is, you can use a goniometer, right? The instrument which is used for measuring the contact angle, to also quantify the surface free energy or surface tension. Okay. The above three techniques, they are not useful for this application. This is another question. What is fatigue basically? It is based on that. So fatigue is a, like, it's better if you guys were, responding. But fatigue is a phenomena that happens into a solid material inside the bulk, in the bulk. So often there are some small microscopic cracks that starts that is maybe there due to manufacturing defect or something like that. If that solid surface is uh, like in an oscillating manner, It is constantly being subjected to stresses. Okay. So in a like this, like stresses increase and then decrease, increase and decrease. So it is oscillating continuously. The same thing is happening. Okay. So what happens? Those cracks, which are initially small, they starts to become bigger and bigger. So if there are multiple cracks nearby, they will start merging. And as a result, after some cycles of stresses, it will break. Okay. So this type of fracture that has happened due to cyclic stresses is called fatigue. So this becomes useful when this kind of cyclic load is always there around the biomaterial. Like when you walk and and a person recently had maybe knee joint replacement. So you're constantly moving your leg forward and backward when you're walking or when you are sitting, and the motion is always similar, and it happens in a like in regular intervals, like up and down, like the movement of the leg. So that is a type of a fatigue load that the material experiences. So we also quantify what is the maximum fatigue load that the material can withstand. So as I have already told, uh, the definition: fatigue is a process by which the structures fail as a result of cyclic load, cyclic stresses. This is called fatigue. Okay. This is the next question. Cytotoxicity of a material or a construct, it can be determined by which of these tests. So cytotoxicity, what this term means cell killing. Cyto means cell, toxicity means poisonous. Right? So it is so toxic that the cell are dying. Okay. So it is cytotoxicity. The cytotoxicity of a material, it can be determined by which of these tests. So first is Ames test, alkaline phosphatase assay, MTT, protein adsorption study. So here we are, so opposite of cytotoxicity will be cell viability. If the material is cytotoxic, then it will result in lowering of cell viability. So basically, you have to then see which of these assays are going to help you quantify cell viability. So we have already seen this name previously in one of the question. Would anyone would like to try? What is the answer, or what could be the answer, correct answer to this question? Anyone? Okay. So the correct answer is MTT. So you can use the same MTT assay that we talked about previously to quantify the cytotoxicity of a biomaterial. The other tests that are mentioned here, it is Ames test. It helps in quantifying the um mutagenicity. Mutagenicity means you want to understand whether your material can cause some mutagenic changes in the genetic material of the cells. Okay, or whether the material is a possible mutagen that can change the genetic response of your cell. So in order to quantify that, we use Ames test. Then this is an assay that is useful to quantify the enzyme alkaline phosphatase. So this enzyme is released, predominantly released in bones, liver. Okay. So as a result of their metabolism, this enzyme is released. So if we are trying to create a bone tissue and you want to validate whether it has successfully been formed, you can quantify whether ALP or alkaline phosphatase is being released or not. Then the fourth one, protein adsorption study. So here we try to see, we expose the material surface to a mixture of protein, all right, and see whether the protein molecules have adsorbed. So there are various adsorption curves, or that is called adsorption isotherms, that are helpful here. Okay, there's one called Langmuir isotherm that talks about that the molecules can only adsorb on the surface in a monolayer, and we can then quantify the characteristics of this monolayer. So that concept is useful when we are trying to quantify how much of the protein that is present in solution has adsorbed on the surface of a material. Why protein adsorption study is useful? Because after all, it is the layer of adsorbed protein which the cells are going to get adhered to. Okay, directly cell and material interaction doesn't take place usually, because the material is in aqueous phase and in physiological environment where there is always proteins available, like plasma proteins and all. They will first directly and interact with the material, and then cells will interact. So that protein layer, the nature of that protein layer will help you understand uh, cell adhesion properties of your material. Okay. Then another question is this: mesenchymal stem cells are what? Are they totipotent, multipotent, pluripotent, or unipotent? Mesenchymal stem cells, also called MSCs. So what is totipotent? Totipotent refers to a cell type which can differentiate along all lineages. Can differentiate along all lineages. Okay, all lineages. So um, totipotent cells, usually the embryonic stem cells. If we know how to do it, we can direct the differentiation of embryonic stem cells along all lineages of cell type that are there in our body. But we still haven't yet understood. Only for a few lineages it is described, but theoretically it is possible with embryonic stem cells. Multipotent means multiple lineages. It can differentiate along multiple lineages. So multiple usually refers to one or two or three lineages, not more than that. Okay. Pluripotent is like refers to those cells which can at least show more than three types of lineage differentiation. Okay. So pluripotent, multipotent, very similar terms, by the way. And also I will say that pluri means plural, more than one lineages it can differentiate. Unipotent, one. So the cell type can only differentiate along one lineage. Okay. So MSCs, or the mesenchymal stem cells, they're actually multipotent in nature. People generally differentiate these cells along osteo, bone cells, chondro, adipo, fat cells. So these three lineages very well you can differentiate these cells. Okay, mesenchymal stem cells. Although more and more literature are coming nowadays that talks about differentiating these mesenchymal stem cells into maybe blood cells and all, but those are very indirect. Generally, and the most easy thing to do is to differentiate these cells along these three lineages. Okay. This is the next question. X-ray photoelectron microscopy. In short, it is also called XPS. Okay. It helps in understanding the following, except means uh, out of this four option, three is correct for XPS and one is not correct. We have to identify the incorrect point. XPS is a technique that again helps in surface characterization. Please note surface characterization. Here we try to understand what are the elements are there on the surface, what is their oxidation states, what is the percentage of these elements present. Okay. So XPS, the instrument is very, very expensive and it's very sophisticated. Hardly any academic institute can afford this instrument. Okay. So it's a very sophisticated, one of sophisticated instruments that is there for characterization of materials. So which of the following option cannot be done by XPS? So I'll directly tell you the answer. XPS can help you to understand the atomic composition. It can help you understand the oxidation states. Also, it can help you give the quantification, quantify the elements that are there. So atomic composition, you can tell what all atoms are there. Okay. Or what are elements are there in your surface. Then at which oxidation state it is, carbon 2 plus, 4 plus, like you can quantify that oxidation state as well. Then also you can quantify the percentage of the carbon in that particular oxygen state. So these very critical information are useful when you are trying to synthesize say new materials and you want to see the effect of the material surface on cell behavior. What it cannot tell you is the wettability of the material, means how hydrophobic or hydrophilic it is. Okay, which is why option D is correct. This is another question. Universal tensile, universal testing machine, universal testing machine, also called UTM. It is a machine that can help you determine the following except. So UTM looks something like this. UTM machine. So there is two ends. These are which are used. So this end and this end. Okay. This is one end, top one. This is another end through which you can hold your sample. And one end is fixed, and to another end there is a, sensor. Okay, that through which you can give some load to your sample of specific value. That is also called load cell. Load cell is connected here. So you can hold your sample in between the two grips, and you can provide some tensile stress or tensile loading, or you can so this instrument or these grips are for tensile loading. Similarly, in UTM, you can do this type of UTMs are also there, where both the ends consist of blunt circular thing, and in between you keep your sample, and both ends are compressed. Okay, put together. So a compression load can be provided here. Then you can also use this machine for, so let me show you. So here what they are doing, the sample is here at the bottom and load is applied at the center so that the horizontal sample piece bends. So some bending can be done. That is called bending test. Getting a very good image. So here I was showing that a sample can be held in between the two grips and uh, provided a tension. Okay. Bending test. Maybe this will be a good image. So here the white colored thing that you can see is the sample, and it is kept in a horizontal fashion, and in the middle of that one tip thing that is coming on top of it, it is applying some load, because of which the material will bend like this. Okay, it will bend like this. That is called bending test. So you can then measure how much load is actually needed to complete the bending. Here, what is the maximum load it is needed to cause bending. So really you can quantify the maximum tensile stress, tensile compression test using this particular instrument. So it can help you quantify these three properties of your material. What it cannot tell you is the surface roughness. Okay, the surface roughness can be quantified through AFM, or atomic force microscopy, which I've already covered. The correct answer here is surface roughness for this question. Then another question is there. Following are the techniques used to measure the porosity of the material, except. Okay. So again, a similar question we have done previously. There is mercury porosimetry, SEM, scanning electron microscopy, gas pycnometry, and dynamic light scattering. Okay, so we have seen that all these three techniques are widely used for measuring the porosity. Here, we saw in the previous question there was an option called liquid displacement method. So, so here, when we are using mercury in that, we are calling that method as mercury porosimetry. But this technique, dynamic light scattering, also called DLS, cannot be used to measure porosity. It cannot be used to measure porosity. This technique is useful to determine the hydrodynamic radius of nanoparticles. So nanomaterials, okay, nanomaterials. So if your material or your particle is very small, like in nanometer range or micrometer range, and you want to know the size of it, you can use DLS technique to quantify it. Then dash detects the vibrational characteristics of chemical functional groups present in the sample. So options are XPS, FTIR, NMR, AFM. So we have already covered what is AFM, what is FTIR, what is XPS. So NMR is also like slightly I have covered. It's again a technique where you are able to measure or find out what are the functional groups, chemical functional groups are there. But here we use magnetism, okay, nuclear magnetic resonance phenomena to quantify it. So since the question talks about vibrational characteristics, means you are causing bonds between the atoms to vibrate, and that happens in presence of IR. So the correct answer here is FTIR. Using FTIR, you can detect the vibrational characteristics of chemical functional groups. And I've already shown you in the beginning the FTIR spectrum, how it would look like. This is another technique. Which technique is used here to understand the thermal behavior of the material? Again, similar question we have done previously. UTM, Universal Testing Machine, scanning electron microscopy, DSC, differential calorimetry, mercury porosimetry. So the correct answer here is DSC. Okay. So DSC is a technique that helps us to understand where is the what is the glass transition temperature, what is the melting point. So we can quantify that. Those temperatures we can find out about a particular polymer, uh, and that basically tells us about the thermal properties of the polymer. Okay. This is another question. Which of the following is not a hemocompatibility study? So hemocompatibility is a fancy way of saying blood compatibility, by the way. Compatibility. Okay. So which is not a blood compatibility test. Hemolysis assay, Ames test, platelet addition assay, protein adsorption assay. Anyone? Okay. So hemolysis assay is a technique or is an experimental technique where you see whether the blood cells are getting lysed in presence of your material, okay, whatever material it is. It is called hemolysis assay. Then Ames test, I already told you, it helps us to quantify the mutagenicity of our material, whether the material can cause some mutational changes, a mutation into the mammalian cells that are going to be um, grown or cultured on this material. Then platelet adhesion assay. So here we're talking about the platelets which are there in the blood and whether or not these platelets can adhere on your surface of the material. Third and the fourth one is protein adsorption assay. So proteins which are there in our bodily fluids like plasma protein and all. So we can quantify whether those proteins are able to get adsorbed on our material. So among these four options, which is not a hemocompatibility study, then is Ames test. Ames test is not a hemocompatibility study. Okay, it has nothing to do with blood. Okay. This is the next question. Surface characterization of a material can be done using which of the following techniques? Thermogravimetric analysis or TGA, universal testing machine, scanning, differential scanning calorimetry, or SEM, scanning electron microscope. Correct answer is SEM. Okay. You subject the sample to some electron beam and you can capture the image of the surface. So these are the some of the questions, around 20 questions I tried to cover in today's session. Okay. If you have any queries related to any question or any topic that you have not understood in today's discussion, let me know. I'll have I'll wait for you guys for 3-4 minutes, and if there are no further questions, then I stop today's meeting. Are there any questions? Well, the questions that which I discussed today, um, have you guys understood them? The message that I tried to convey through every slide, was I successful in um, conveying it to you properly? Like, means, did you have any doubt meanwhile I was explaining? Please feel free, like chill. You can definitely ask me and I'll help you. That is why I'm here today.
No doubt, ma'am. It was clear.
Yeah. Okay. So I hope after this you guys will watch the lectures of week 4. Okay. The things will become much more clear and attempt your assignment well. Okay. Try to get all the answers correct. Okay. Then thank you all for joining. I'll end today's session and I'll see you guys next week, Saturday at 4 p.m. Okay.